Biology Part 84. The transfer of fluid into the kidney tubules is assisted by a rather unusual cellular arrangement at the junction of epithelial cells of Bowman's capsule and endothelial lining cells of the blood capillaries. The detailed structure of this junction is illustrated in the figure. As you can see that the endothelial cells of the capillaries are perforated by tiny pores, which allows the blood to come into the direct contact with the underlying basement membrane. This membrane forms only barrier between the blood and the cavity of the nephrons. For the epithelial cells and podocytes of Bowman's capsule are structured so as to allow the free passage of the filtrate between their foot-like processes. The basement membranes acts as the selective barrier, allowing the water-insoluble substances with the relative molecular mass of up to 68,000 to cross. Therefore, all components of blood plasma except protein molecules pass through into the nephron, forming the fluid called glomerular filtrate, and we can see it in the table as well. Reabsorption by kidney tubules. The two kidneys of an adult human filter about 60 liters of blood per hour, produce glomerular filtrate at a rate of approximately 7.5 liters per hour. Within the same period, only about 0.07 liters of urine is formed, a volume which corresponds to less than 1% of the glomerular filtrate. This, at least, is 99% of the water present in the filtrate is reabsorbed from the kidney tubules back into the bloodstream. A comparison of the composition of glomerular filtrate with that of blood plasma and urine shows that many solutes are likewise reabsorbed. On the other hand, some substances are concentrated by passage through the kidney tubules and they are removed from the body when urination occurs. Reabsorption of the solutes. Glucose present in the glomerular filtrate is actively reabsorbed in the next section of nephron and called the first convoluted tubule. Active reabsorption in this region is facilitated by the structure of epithelial lining cells of the tubule. These cells have a surface membrane in microvilli, which increases the surface area in contact with the tubule contents, and contain numerous mitochondria capable of supplying energy. The reabsorption process involves specific carrier molecules located in a membrane of the epithelial cells, and glucose is transported to the opposite side of the cells where it is taken by the blood capillaries surrounding the tubules. Glucose is reabsorbed completely in normal circumstances and only appears in the urine when its concentration in the blood plasma exceeds the kidney capacity for reabsorption, as is in the case of the disease diabetes mellitus. Sodium ions are also actively absorbed from the first covulated tubule. In this case, reabsorption is less complete but still accounts for up to 90% of the sodium ions present to glomerular filtrate. As sodium ions are pumped out, they carry them an equal number of negatively charged anions, and other ions actively reabsorbed in the first covulated tubule include potassium K+, phosphate H2PO4 negative, and hydrogen carbonate HCO3 negative ions. Most of the remaining sodium ions are reabsorbed in the other region of the tubule, so that 540 grams per day typically present in the glomerular filtrate. Only about 3 grams per day is excreted. The major of the urea present in the glomerule filtrate remains within the tubule, but some is lost and diffuses positively outward. Some substances, including a number of toxins and drugs such as penicillin, pass across the epithelial cells in the opposite direction. That is, they are transported from blood capillaries into the tubule for eventual excretion.